Hello students, we are going to see today dissection of a hibiscus flower. Now you all know that hibiscus flower we have to dissect into 4 parts: calyx, corolla, androecium, gynoecium. Before going ahead, I want to tell you, you have to take the flower. You can do and you can try it at your home also. You have to take the flower which is of 5 petals. Then only you will get the correct number of calyx, corolla, androecium and gynoecium. If you take hybrid variety which is made up of number of petals, then accordingly when you will take the section of the ovary also it will change. So it's better, I will suggest that you will take a flower which has 5 petals. So find such flower which has a 5 petals. Now let's go ahead with the dissection of a flower. Now since it's a dicot plant, hibiscus plant is a dicot plant, we all know the distinguish between monocot and dicot. That is, it shows dicot plants flower shows pentamerous symmetry. So we have all parts of a flower in terms of or in multiples of 5 or 5 in number or multiples of 5. So let us see, this is a stalk or the pedicel of a flower. This is a stalk or the pedicel of a flower. Now what I am showing it to you, I'll turn the camera now. Friends, we all know that we are in the lockdown period, so we don't have at home slides and cover slip and all the materials. So whatever materials are available with me, I am trying to show the dissection to you. Now this is a pedicel as I told earlier only. Now I am removing this, I don't have forceps also at home, so I am removing this epicalyx which is you can see here a small thing over here which is green in color, which is epicalyx. So this is the epicalyx. I am removing it. Now I will stick it afterwards on the piece of paper so that you can understand different parts of flower very easily. Now I have removed this epicalyx, you can see it over here. Now I am removing calyx which is made up of 5 sepals. Now you can see this 1, 2, 3, 4 and 5. So which is made up of calyx which is made up of 5 sepals which are usually green in color. In this hibiscus flower these are green in color. Now I am removing corolla which is made up of 5 petals. Usually it is colorful. In this flower it is again colorful, that is red in color. This is also 5 in number. So this is corolla which is again 5 in number. 1, 2, 3, 4 and 5. So all of you can see this 5 in number. Now I am removing androecium and gynoecium. Now you can see this here, androecium which is here in the staminal tube, and here you can see these, you can see here stamens are here which contain pollen grains inside this. Now you can see here in case of this hibiscus flower pentaphyid stigma. Stigma is of 5 parts. So pentaphyid stigma. So I am going to dissect it now. Now slowly from the side part without damaging the ovary, I am trying to remove it. This is androecium I am removing. Now you can see this white line. This is a white line which is a part of the style. Now I have removed the style and stigma I have separated out. You can see it. I am removing this part slowly. So this is a androecium I have removed on the paper, you can see, which is of stamens and the anthers. Anthers are having the pollen grains inside them. Now you can see there are number of anthers, innumerable number, and slowly I am trying to remove this. You can see this cover, I am trying to remove it. So this is ready. This is ovary which is at the base, then style and pentaphyid stigma. You can see it clearly, ovary. This is ovary, style and stigma. Stigma which is pentaphyid. So I am going to stick this on the piece of paper and then I'll show you all the parts. So epicalyx, then calyx which is made up of 5 sepals, corolla 5 in number, made up of petals, 5 petals, then androecium and gynoecium, ovary, style and stigma. This is stigma. So with this we have completed the dissection of a flower. Now I'll stick it on the piece of paper and I'll show you the all parts. Now see this all parts: epicalyx, calyx, corolla, androecium and gynoecium, pedicel or stalk, ovary, style and stigma.